Hello my lovely students, welcome to Infinity Learn by Sri Chaitanya. This is Dr. Priyanka and today we are going to discuss previous year questions of Biotechnology Principles and Processes. Hope you all are ready for today's class. आप सभी तैयार हो आज के इस क्लास के लिए? As you know, in this series हम लोग सारे chapters के previous year questions discuss कर रहे हैं बच्चों, right? So hope you all are ready with high josh and your pen, pencil, everything, notes and everything, okay? And before starting today's class, I would like to tell you how you can register for our starter package, right? कल भी बताया था मैंने, your starter package आपको मिल रहा है 499 रुपीज़ में. You are getting 70% discount on this starter package. तो कैसे आपको वो avail करना है, that I will tell you. You will have to go to our description box, ठीक है? जहाँ आप मेरा वीडियो देख रहे हो, नीचे आपको मिलेगा description box. This is our description box. यहाँ पे आपको सारे इंपॉर्टेंट लिंक्स मिल जाएंगे बच्चों. शो मोर पे हम क्लिक करेंगे. ओके, सो यू कैन सी इन्फिनिटी लर्न फ्री मास्टर क्लास के लिए आपको एक लिंक नजर आ रही है यहाँ पर. टेलीग्राम चैनल के लिए भी ऑल्सो यू हैव लिंक हियर. देन फॉर इम्पॉर्टेंट प्ले लिस्ट ऑल्सो यू विल हैव लिंक्स हियर. ओके, एंड ऑल दीज लिंक्स आर very helpful. So let's understand and let's see how you can register for our Infinity Learn Masterclass in just rupees 499. And what is this? That also I will tell you, okay? So you will click on this link, okay? By clicking on this link, you will come to our web page. इस तरह से आपको पेज नजर आएगा, right? नीचे स्क्रॉल करना है आपको. एंड जिस क्लास में आप पढ़ रहे हो, जिस पैकेज को आपको सब्सक्राइब करना है, उसको आप सिलेक्ट कर सकते हो. इफ़ यू आर प्रिपेयरिंग फॉर जेई और इफ़ यू आर प्रिपेयरिंग फॉर नीट, राइट? सो इनफैक्ट इफ़ यू आर इन जूनियर क्लास, आप क्लास देख रहे हो 6th 7th 8th 9th 10th 11th 12th सब के लिए अवेलेबल है बच्चों ये पैकेज. ओके, सो आई विल क्लिक ऑन नीट फर्स्ट. नीट पे क्लिक किया, देन यू विल सी द सब्जेक्ट दैट इज कवर्ड विल बी फिजिक्स, केमिस्ट्री, बायोलॉजी. तीनों सब्जेक्ट जो है कवर किए जाएंगे in this package. And in this course you will get 3 mock tests, you will get 14 previous year question papers, then 1000 plus self-learn videos, 1000 plus self-learn flashcards, 3000 plus adaptive practice questions and also doubt resolution. And all these things you are getting in just rupees 499 dear students, and this you are getting at a discount of 70%. ऐसा मौका बार बार नहीं मिलेगा. ओके, फटाफट से आप रजिस्टर कर लो इस कोर्स के लिए. एंड द वैलिडिटी ऑफ दिस पैकेज विल बी फॉर 1 month. ओके, एंड होप दिस पैकेज विल बी वेरी यूजफुल. ओके, ऊपर जाके आप यू कैन गो हियर एंड यू कैन fill your details here. You will have to write your first name, last name, your email ID. Then you can select your course, your grade, whether it is JEE, NEET, 11th, 12th, that you can select here. And then you can enter your phone number and submit. Okay. So hope guys you have understood that how you can register for our Infinity Learn Master class. मैं हमेशा बोलती थी डिस्क्रिप्शन बॉक्स में आपको लिंक मिलेगा. टेलीग्राम चैनल ज्वाइन करो, आपको लिंक मिलेगा. अब लिंक कहाँ मिलेगा ये समझ में आ गया, राइट? ओके, सो लेट्स स्टार्ट विद आवर टुडेज सेशन. सो आवर टुडेज सेशन इज ऑन बायोटेक्नोलॉजी प्रिंसिपल्स एंड प्रोसेसेस previous year questions. Okay, so we will be studying the previous year questions of this chapter, right? Don't forget to subscribe Infinity Learn NEET channel. अगर आपने अभी तक सब्सक्राइब नहीं किया है तो अपने सारे जो भी आप देख रहे हो इतने सारे प्लेलिस्ट है. We have given you so much nice sessions, so please subscribe on Infinity Learn NEET channel, okay, on YouTube. And also, like I told you, you can subscribe for you can register for our NEET Infinity Learn Masterclass. How you are going to access this link? I told you by going to the description box. So let's start with our first question of Biotechnology Principles and Processes. Let's start. So what is the first question here? Let's understand this question was asked in NEET 2017 में पूछा गया था ये question. क्या है ये question? The DNA fragments separated on an agarose gel can be visualized after staining with. Okay, so they are talking about gel electrophoresis. Okay, gel electrophoresis will help us to segregate the fragments of DNA, okay, based on their molecular weight. Okay, so the fragments of DNA that will have less molecular weight will move further, and ultimately you will get bands of DNA in the agarose gel. But you cannot see those bands with the naked eyes. We will need to use a stain. We will have to stain these DNA fragments to be able to see them, right? So they are stained by, anybody in the chat? They will be stained by ethidium bromide dye. The DNA fragment should be stained with ethidium bromide dye, and then we can visualize that in the gel. So here you can see, can you see the bands? These are the bands of DNA that is stained by ethidium bromide, and we can see them in the UV light. UV light में हम इनको देख पाएंगे after we have stained them with ethidium bromide. Okay, so this is how your DNA fragments will look in the agarose gel. Okay, dear students, so this was about first question that was asked in the exam NEET 2017. Now let's see the next question on the screen. DNA fragments are negatively charged or neutral, either positively or negatively charged depending on the size, or positively charged. Question that was asked in again NEET 2017. So biotechnology principles and processes ये ऐसा chapter है जहाँ से maximum questions पूछे जाते हैं. 4 to 5 questions can also be asked from this chapter, right? So this is a very important chapter. Let's understand how questions are asked in the exam. So now they are asking what is the charge on DNA. So your DNA is negatively charged. Your DNA is negatively charged because of the phosphate group. Okay, and we use this characteristic of DNA to separate it in the agarose gel in gel electrophoresis. So what we do, very good, A is the correct answer, Bardaval. Now what we do is, in gel electrophoresis there are wells, okay, here, and this tray is filled with agarose gel, right? Now in this well we will keep our fragmented DNA, and then the ends will be attached to a energy source, right? Since our DNA is negatively charged, this end will be positively charged and this end will be negatively charged. So our negatively charged DNA will start moving towards the anode end. This is the anode end and this is the cathode end, right? So DNA will start moving from cathode to the anode end because it is negatively charged. It will be attracted to the positive end, right? And the molecule of DNA that is smaller will be able to go further, right? And the molecule of DNA that is heavy will not go that far, isn't it? So this is how we are going to separate the fragments of DNA and we will get bands. Okay, so this was about gel electrophoresis. Correct option for this question will be A. Okay, A. And again, when these DNA fragments are separated, we need to dye them with ethidium bromide dye and then we can see the DNA in the UV light. Okay, तभी हम देख पाएंगे जब हम UV light को expose करेंगे the stained DNA. Okay, now this was your second question. Correct option was A. Yes, very good, Bardaval. Now let's move on to our next question. A gene whose expression helps to identify transformed cell is known as vector, plasmid, structural gene or selectable marker. This question was asked in NEET 2017. That gene that will help us to distinguish between transformed and non-transformed cell, right? So that is known as selectable marker, जो हमें help कर रहा है select करने में transformed and non-transformed cell को. We will see the whole process, don't worry. But correct option for this question will be selectable marker. Let's understand how. What is this? This is a plasmid. The name of this plasmid is PBr322. Please understand carefully. This is very important and this could be asked in your exam in NEET 22. So this is our plasmid PBr322, and this plasmid, can you see here, ampicillin resistant gene and tetracycline resistant gene. Okay. Now these are the genes that is responsible to make the bacteria resistant to a particular antibiotic, and those antibiotics are ampicillin and tetracycline. If this gene is present, dear students, then only your bacteria will be resistant to tetracycline and ampicillin antibiotics. Okay. Now what will happen is, if I will insert a gene of interest in this gene, okay, so in this gene, ampicillin resistant gene, from here to here, you can see there are two restriction sites. What are these? These are restriction sites. So this PST1 and PVU1 are the restriction sites which are present in this gene, ampicillin resistant gene. Okay. If I cut this gene at this restriction site and insert my desired DNA into this, then this gene will not be able to work. Why? Because its integrity is lost, right? अगर मैं इस जीन के बीच में इंसर्ट कर दूं अपना जीन ऑफ इंटरेस्ट और डीएनए ऑफ इंटरेस्ट, जिसके मैं मल्टीपल कॉपीज चाहती हूँ, व्हाट विल हैपन? दिस जीन विल नॉट वर्क. That means your bacteria in which this plasmid is there will not be able to resist against this antibiotic, that is ampicillin. Ampicillin resistant gene को अगर मैंने बीच में से कट करके अपना gene of interest डाल दिया, now that gene will not be helpful. Now this bacteria is not resistant to ampicillin. Similarly, this gene, that is tetracycline resistant gene, अगर मैंने यहाँ पर insert कर दिया अपना gene of interest, then what will happen? Again your bacteria will not be resistant to tetracycline now. And by growing those bacteria in the medium containing ampicillin and tetracycline, now I can figure out that which are transformed and which are not transformed. So these genes are known as selectable markers. So what are selectable markers? Let's write it down. Selectable markers are ampicillin and tetracycline resistant gene in plasmid. Okay. So in this plasmid, in this particular plasmid, these genes are selectable markers. There are more selectable markers. We will study them one by one in our session. Okay. So the correct option for this question was very simple. Option D, selectable markers. Hope you all have understood. Okay. Now let's move on to our next question. What is the criteria for DNA fragments movement on agarose gel during gel electrophoresis? Many a times questions from gel electrophoresis are asked. All those who are watching this session, please tell me hi, hello in the chat and give answer for this question. What will be the correct option? अभी हमने discuss किया. In a gel electrophoresis, the DNA fragments will move according to their molecular weight. जितना कम molecular weight होगा उतना आगे जाएगा, isn't it? जितना लीन होता है हमारा बॉडी, हम उतने एक्टिव होते हैं, एनर्जेटिक होते हैं, उतने ज़्यादा दूर तक हम लोग भाग सकते हैं, राइट? सो सेम इज द केस विद DNA हियर. So let's see the statements. The smaller the fragment size, the farther it will move. Yes, that is correct. जितना small size होगा DNA fragment का, उतना दूर वो जाएगा in our gel medium. Positively charged fragments move to farther end. Nothing like this. Our DNA is negatively charged, so this is wrong. Negatively charged fragments do not move. Yes, they move. They are attracted towards the other end, positively charged end, which is known as anode. Just now we have discussed, right? The larger the fragment size, the farther it moves. No. The smaller the fragment size, the farther it will move. जितने हम lean, thin, active होंगे, उतना ही ज़्यादा हम आगे जा पाएंगे. अगर हमने weight gain किया, तो it will be difficult to move that far, isn't it? So इसलिए ये वाला जो है ऑप्शन गलत होगा. करेक्ट ऑप्शन फॉर दिस क्वेश्चन इज ए. अंडरस्टूड डियर स्टूडेंट्स? हाय शिव. चलो, नाउ लेट्स मूव ऑन टू आवर नेक्स्ट क्वेश्चन. द प्रोसेस ऑफ सेपरेशन एंड प्योरिफिकेशन ऑफ एक्सप्रेस्ड प्रोटीन बिफोर मार्केटिंग इज कॉल्ड. ठीक है, व्हाट वी हैव डन, हमारा प्रोडक्ट बनके तैयार है. ओके, हमारा प्रोडक्ट बनके तैयार है. बड़े बड़े फर्मेंटर्स में जो है हमने हमारा प्रोडक्ट बना लिया है विद द हेल्प ऑफ बैक्टीरियाज एंड एवरीथिंग. नाउ आई वांट माय प्रोडक्ट टू बी सेपरेटेड फ्रॉम रेस्ट ऑफ द थिंग्स एंड ऑल्सो आई वांट टू मार्केट माय प्रोडक्ट. सो दैट प्रोसेस ऑफ प्योरिफिकेशन एंड सेपरेशन of the product that I need to market is known as downstreaming process. Downstreaming process. Okay? बायोटेक्नोलॉजी के हेल्प से हमने प्रोडक्ट तो बना लिया है फर्मेंटर में, ओके, बायोरिएक्टर्स में. अब मुझे क्या करना है, अब मुझे उसको सेपरेट करना है, अब मुझे उसको प्यूरिफाई करना है. सेपरेट और प्यूरिफाई करके ही तो मैं मार्केट कर पाऊंगी, राइट? सो दैट प्रोसेस इज नोन एज डाउनस्ट्रीमिंग प्रोसेस. Okay, so this question was asked in NEET 2017 and this question is asked many times in your exam, many times. Okay, so let's see what is this upstreaming process and bioreactors and what is the downstreaming process. Let's understand. Okay, everyone, very important. So what happens in upstreaming processing? What we will do, we will कंस्ट्रक्ट अ वेक्टर विथ आर डिजायर्ड जीन. जिस जीन का एक्सप्रेशन मैं चाहती हूँ, अगर मुझे कुछ प्रोटीन बनाना है विद द हेल्प ऑफ दैट जीन, सो दैट जीन दैट इज नोन एज जीन ऑफ इंटरेस्ट, दैट GOI I will insert in this vector, and then ultimately what I will do, I will, if I have identified the particular bacteria or organism from which I will get my protein, I will grow those bacterias in a bioreactor. Okay, in a bioreactor. Here the process is fermentation. Okay, so after growing my bacteria in the bioreactor, what will happen? My bacteria will produce the desired protein. Okay, now this protein I will have to, after this protein is formed in this media, what I need to do is I need to extract that protein from the cells and I need to purify that product, right, that protein, and this is known as downstreaming process. So separation and purification of the desired product before marketing is known as downstreaming process. So correct option was A. Okay बच्चों, चलो नेक्स्ट क्वेश्चन पे आते हैं. Stirred tank bioreactors, large huge tanks in which media will be there and then there will be cells and you will provide each and every environment, all the optimum conditions for those cells to grow. Okay, and this happens in large tanks which are known as bioreactors. So they are talking about stirred tank bioreactors have been designed for. नाम से समझ में आ रहा है stirred tank, that means we are stirring, okay, the media in the tank. So you have 4 statements. Let's understand all these statements one by one. प्यूरिफिकेशन ऑफ प्रोडक्ट्स, स्टर्ड टैंक बायोरिएक्टर्स में क्या प्यूरिफिकेशन ऑफ प्रोडक्ट होता है? शिव बोल रहा है सी, देखते हैं क्या है करेक्ट आंसर। एडिशन ऑफ प्रिजर्वेटिव्स टू द प्रोडक्ट, अवेलेबिलिटी ऑफ ऑक्सीजन थ्रू आउट द प्रोसेस, ensuring anaerobic condition in the culture vessel. Now stirred tank है, so obviously there will be availability of oxygen throughout the process. We want oxygen for the cells and we need this oxygen to be distributed evenly in the whole culture medium. So उसके लिए हम use करते हैं stirred tank bioreactors. Here you can see there are stirrers. This is an image of bioreactors. These stirrers are like pedals. इससे सारा मीडिया जो है प्रॉपरली मिक्स होता है एंड उसके साथ में मिक्स होता है न्यूट्रिएंट्स, के साथ में मिक्स होता है ऑक्सीजन. ओके, सो एवरी सेल विल गेट equal amount of nutrients and oxygen, right? Here you can see this is the feeding pump from which you can introduce more media in this bioreactor. Then you will see air also you will pump through the aerator, right? So this process is aerobic process. This is a reactor tank and then there is thermal jacket as well to maintain the optimum temperature. As you know, bacteria and other cells will grow when they will get optimum environment, right? Now this is an example of stirred tank bioreactors. Hope this is clear. Yes, correct option for this question was C. Very good. Next, a foreign DNA in plasmid cut by the same restriction endonuclease. Okay, सबसे पहले तो ये समझते हैं what are restriction endonucleases. Restriction endonucleases are the molecular scissors, right? They are molecular scissors. So we'll write it down. Molecular scissors, right? Now when this molecular scissors are used, the same molecular scissors are used in the DNA and for cutting the plasmid, it can be joined to form a recombinant plasmid using. बहुत सिंपल सा क्वेश्चन है, ये पूछ रहे हैं कि हम तो डीएनए को कट तो कर देंगे विद द हेल्प ऑफ रेस्ट्रिक्शन एंडोन्यूक्लियस, उनको ज्वाइन कैसे करेंगे? ज्वाइन करेंगे बाय द हेल्प ऑफ मॉलिक्यूलर ग्लू. ओके, एंड वो मॉलिक्यूलर ग्लू कौन सा है? दैट इज डीएनए लाइगेज, राइट? मॉलिक्यूलर ग्लू इज ligase. Very good. Okay, molecular scissors are restriction endonuclease. Right? So the correct option for this question will be DNA ligase. Very good. Okay. Now what is Taq polymerase? We will see. Question is asked on Taq polymerase as well. This Taq polymerase is used in PCR technique for polymerization, right? And this Taq polymerase is isolated from Thermus aquaticus bacteria that can withstand very high temperature. बहुत ज़्यादा temperature पर भी ये जो एंजाइम है ये डीनेचर नहीं होता है in PCR. हम लोग देखेंगे, वो भी देखेंगे. Okay? Now let's move on to our next question. Correct option for this was D. Which of the following is not a component of downstreaming processing? Downstreaming process हमने देखा separation and purification. 4 options आपको दिया है, separation, purification, preservation and expression. Preservation भी कहीं ना कहीं will be helpful in downstreaming process, right? Separation तो है ही, purification भी है, preservation is also required before we market. We cannot market our product like this. We need to add preservatives, right? For long shelf life. So A, B, C is there in downstreaming process. What is not there is expression. Expression of the genes, that is formation of the product from the gene, is a process of upstreaming, right? So downstreaming process में क्या include नहीं होगा? क्या include नहीं होगा? Expression, isn't it? Separation is there, preservation is there, purification is there, preservative needs to be added. to be added before marketing. Fine, everyone? Okay, correct option here will be D, dear students. Understood why it will be D? D क्यों होगा, समझ में आया ना? क्योंकि एक्सप्रेशन तो अपस्ट्रीमिंग प्रोसेस है. एक्सप्रेशन मतलब क्या? एक्सप्रेशन मीन्स डीएनए से बनेगा आरएनए ट्रांसक्रिप्शन, ट्रांसक्रिप्शन के बाद होगा ट्रांसलेशन, दैट मीन्स आरएनए विल फॉर्म द डिजायर्ड प्रोटीन एंड दिस इज व्हाट इज एक्सप्रेशन. ओके, सो करेक्ट ऑप्शन हियर वाज डी. चलो, so again the same process, upstreaming, fermentation and downstreaming processing that we have discussed. Next question. Next question on your screen. Which of the following restriction enzymes produces blunt ends? Okay, now blunt ends are opposite of sticky ends. If we are getting ends like this, these are known as sticky ends. These are known as sticky ends. और कहाँ मिलते हैं, कब मिलते हैं? Whenever we cut our DNA, if it is cut at two different sites, then we will get a sticky end. If my DNA is cut from at the same site on both the strands, then I will get a blunt end. अगर ऐसा मिलता है मुझे DNA fragments after exposure to restriction endonuclease, then it is known as blunt end. समझे? अगर मेरा रेस्ट्रिक्शन एंजाइम जो है वो दोनों डीएनए स्ट्रैंड को डिफरेंट लोकेशन पे कट करेगा तो मुझे मिलेगा स्टिकी एंड. और अगर मेरा रेस्ट्रिक्शन एंडोन्यूक्लिएज डीएनए स्ट्रैंड्स को सेम ही साइड पे कट करेगा, देन आई विल गेट अ ब्लंट एंड. यूजुअली द स्टिकी एंड इज प्रिफर्ड, राइट? अब यहाँ पर यू आर गेटिंग 4 ऑप्शंस, यू आर गेटिंग 4 रेस्ट्रिक्शन एंडोन्यूक्लिएज. यू विल हैव टू टेल मी विच वन विल गिव यू blunt ends. They are asking about blunt ends. Okay, option is B, that is fine, eco R5. But let's see what are the different types of restriction endonucleases and what are their restriction sites and whether they are giving us a sticky end or a blunt end. ठीक है, चलो let's see one by one. I will mark the important ones. जो भी important है वो मैं मार्क करूंगी, उसको ध्यान रखना है. आप चाहिए तो you can take a screenshot from here. We will start with eco R1. eco R1, okay. इसका recognition site है GAATTC CTTAAG, okay, and nix will be made between GA and AG like this. Okay, तो जो मैं मार्क कर रही हूँ बस वही ध्यान में रखेंगे, हम सारा नहीं पढ़ेंगे. Then eco R2, the restriction site is CCWGG GGWCC, that's it. Here also we are getting sticky ends. Now coming to the eco R5, very important, जो यहाँ पूछा गया है एग्जाम में, 2016 का क्वेश्चन है. eco R5 में देखो, restriction site is GATATC CTATAG एंड जो निक्स हो रहा है ना वो बीच में हो रहे हैं इस तरह से, दोनों ही स्ट्रैंड में सेम ही स्पॉट पे दिस रेस्ट्रिक्शन एंडोन्यूक्लियस इज कटिंग द डीएनए. सो GAT के बाद यहाँ पे CTA के बाद देयर इज अ निक, दैट मीन्स वी आर गेटिंग स्टिकी एंड्स, इजंट इट? वी आर गेटिंग स्टिकी एंड्स. तो ये याद रखना है आपको, ये 3. देन BAM-H1 इज ऑल्सो इम्पॉर्टेंट. We get BAM-H1 from Bacillus amyloliquefaciens bacteria, right? यहाँ पर भी we are getting sticky ends, no blunt end, no blunt end. Okay, blunt end हमें कहाँ मिला? Blunt end हमें मिला eco R5 में. Okay, then SAW1 Staphylococcus aureus से मिलेगा और यहाँ पर भी हमें मिल रहा है sticky end. तो blunt end हमें कहाँ मिला? सिर्फ eco R5 में. ठीक है, सो द करेक्ट ऑप्शन फॉर दिस क्वेश्चन विल बी बी. जो मैंने मार्क किए हैं बस उसी के रेस्ट्रिक्शन साइट्स आपको याद रखना है, सबसे ज्यादा पूछा जाने वाला यही है, EcoR5, EcoR1 एंड BAMH1। फाइन बच्चों, चलो now let's move on to our next question. Which of the following is not a feature of plasmid? So plasmid होता क्या है पहले तो वो समझते हैं हम। ओके बारदवल, समझते हैं? शिव, what is a plasmid? Plasmid is a double-stranded extra chromosomal circular DNA in a plasmid, in a bacteria. Okay, so usually bacteria will have plasmid. Now what is plasmid? Let's understand once again. Plasmid is extra chromosomal, मतलब जो नॉर्मल क्रोमोसोम्स होते हैं बैक्टीरिया के, विच इज नोन एज न्यूक्लियोइड, उसके अलावा दिस इज प्रेजेंट। ओके, सो एक्स्ट्रा क्रोमोसोमल, डबल स्ट्रैंडेड, डबल स्ट्रैंडेड, एक्स्ट्रा क्रोमोसोमल, डबल स्ट्रैंडेड, सर्कुलर, सर्कुलर होता है ये, सर्कुलर डीएनए, प्लाज्मिड। नाउ व्हाट इज द मीन्स यूज ऑफ दिस प्लाज्मिड? यस, वेरी गुड बारदबल। व्हाट इज यूज ऑफ दिस प्लाज्मिड? दिस प्लाज्मिड कैन रेप्लिकेट इंडिपेंडेंट टू द न्यूक्लियोइड। सो बाकी के जेनेटिक मटेरियल के अलावा ये जो प्लाज्मिड है, इट कैन रेप्लिकेट बाय इटसेल्फ। Okay, and that site is known as origin of replication site from which the replication will start. Okay, so this plasmid can replicate independently. Let's write it down. It can replicate independently because it has origin of replication site, ORI site. If I attach my desired gene of interest to this replication site of plasmid, what will happen? My gene of interest will also be replicated, right? And with the help of this plasmid, I can transfer my gene of interest in the other organism or other cell or other bacteria. So very good, Shiv. This vector, this plasmid will act as a vehicle. It will take my desired gene, it will take my gene of interest to the next bacteria, right? So very good. So this is about plasmid. Now what is not the feature of plasmid? They are asking this only, right? Which of the following is not a feature of the plasmid? चार ऑप्शन आपके पास है। ट्रांसफरेबल, यस इट इज ट्रांसफरेबल, तभी तो हम ट्रांसफर करेंगे हमारा जीन ऑफ इंटरेस्ट। सिंगल स्ट्रैंडेड, नो इट इज डबल स्ट्रैंडेड ना, इट इज डबल स्ट्रैंडेड एंड नॉट सिंगल स्ट्रैंडेड। इंडिपेंडेंट रेप्लिकेशन, एब्सोल्युटली करेक्ट। व्हाई? बिकॉज आई टोल्ड यू दे हैव ओरिजिन ऑफ रेप्लिकेशन साइट, दे कैन रेप्लिकेट ऑन इट्स ओन। एंड सर्कुलर स्ट्रक्चर, यस दे आर सर्कुलर। ओके, एक एग्जाम्पल हम देखते हैं, PBr 322, दैट इज वेरी वेरी इंपॉर्टेंट। करेक्ट ऑप्शन फॉर दिस क्वेश्चन विल बी बी इटसेल्फ, बी। ओके, चलो नेक्स्ट इज टैक पॉलीमरेज। टैक पॉलीमरेज, दिस एंजाइम is used in PCR, which is also known as polymerase chain reaction. Okay, we will get many copies of our DNA with the help of this PCR. Now question is very good, Shiv, देखते हैं क्या है correct option. Taq polymerase is obtained from. Now remember, in PCR temperature will go high. Not all enzyme can sustain such a high temperature, right? So this Taq polymerase is what? This is this is resistant to temperature. Okay, it is resistant to higher temperature. So अगर temperature बढ़ भी जाता है ना, this enzyme will not get denatured. Enzymes are protein ना, mostly, yeah. There are ribozymes as well. Those are nucleic acid, right? So there are 4 names of bacteria: Bacillus subtilis, Pseudomonas putida, Thermus aquaticus, Thiobacillus feroccidens. Correct option is Thermus aquaticus bacteria. Now this Thermus aquaticus bacteria is present in hot sulfur springs. Okay. So naturally this bacteria is present in hot sulfur springs. That means the enzymes in this bacteria will be resistant to high temperature. So we get our Tech polymerase from Thermus aquaticus bacteria and use it in our PCR, that is polymerase chain reaction. Simple. Correct option here was C. Very nice. Don't forget to like and share this session बच्चों. सेशन लाइक भी करो, शेयर भी करो अपने फ्रेंड्स के साथ में, एंड सब्सक्राइब करना मत भूलो इन्फिनिटी लर्न नीट चैनल को. ओके, नेक्स्ट. अरे, which of the following is a restriction endonuclease? 4 option: DNase 1, RNase, Hind2 and protease. NEET 2016 का क्वेश्चन है. Restriction endonuclease are what? These are molecular scissors. Which are used to cut DNA. Fine. Then you have options: DNase 1, RNase, Hind2 and protease. The correct option is C, that is Hind2. This restriction enzyme we get from Haemophilus influenza. Haemophilus influenza is a bacteria, not a virus. So we get this Hind2 from Haemophilus influenza bacteria. Fine. Next. Which of the following is not required for any of the techniques of DNA fingerprinting available at present? So here they are talking about the techniques of DNA fingerprinting. Restriction enzyme, DNA hybridization, polymerase chain reaction or zinc finger analysis. Okay, let's understand what is DNA fingerprinting. See, there are unique sequence of DNA in each and every individuals, but the DNA sequence of a child may be similar to the DNA sequence of the parents, right? Similarity is there, but again individual to individual it will differ, right? So what we will do is we will perform gel electrophoresis after using restriction endonuclease on the DNA sample, and we can see the similarity, right? Now what technique is not used? They are asking, not used in DNA fingerprinting will be the zinc finger analysis. This has nothing to do with DNA fingerprinting, no. Restriction enzymes are required. Restriction enzymes are molecular scissor. We need to cut the sample of DNA. We will use for that restriction enzymes. DNA hybridization is also important. Why? Because I will need to make multiple copies of the sample that I have. राइट मेरे पास एक छोटा डीएनए का सैंपल है, आई वांट इट टू एम्पलीफाई. ओके, बिकॉज मैं जब ये पूरा प्रोसेस करूंगी जेल इलेक्ट्रोफोरेसिस एंड एवरीथिंग, डीएनए फिंगरप्रिंटिंग, आई विल नीड मल्टीपल कॉपीज ऑफ दैट डीएनए फॉर दीज प्रोसेसेस. So what I will do PCR, polymerase chain reaction, and I will make multiple copies. For that I will need this DNA hybridization, and also polymerase chain reaction is required in DNA fingerprinting. एक single DNA fragment को I cannot use for multiple processes. No, I need to amplify those. Number of DNA should be more. Okay. So polymerase chain reaction is also there. Now this zinc finger analysis is nowhere in our DNA fingerprinting. No. Correct option was D. Next question. The DNA molecule to which the gene of interest is integrated for cloning is called. Cloning means, what is cloning? Cloning means to make copies, multiple copies. Okay. Now for making multiple copies, I need to insert my gene of interest inside a plasmid so that it can make its copies, and also with the help of this plasmid I will be able to transfer my gene of interest inside the host cell. So this is my gene of interest. This is my gene of interest and this is my plasmid. Okay. So if I want my gene of interest to be carried to the host cell, I will need what? Template, carrier, transformer or vector? Vector. Very good. D. I will need a vector, right, to carry my gene of interest inside the host. Okay, that will act as a carrier. Fine. Now let's move on to our next question. The cutting of DNA at specific locations became possible with the discovery of. देख रहे हो घुमा फिरा के they are asking questions on the same concept, but repeatedly they ask questions on this concept. Okay. For cutting of DNA, what do you need? For cutting of DNA, what do you need? Tell me in the chat fast, everybody. What will I need to cut my DNA? And also my plasmid. Yes, I will need restriction enzymes, which are also known as molecular scissors. Okay, like EcoR1, EcoR5, BAM-H1, HIND3, HIND2. There are many. Yes, restriction enzymes are molecular scissors that will help in cutting of the DNA. Selectable markers क्या होता है? We have seen in the first question. Selectable markers are those genes which are present in your plasmid that will be resistant to a particular antibiotic. किसी एक antibiotic के लिए वो resistant होंगे. That means when I will cut that gene and I will insert my gene of interest in that region, what will happen? That gene will become non-functional. Now my plasmid or my bacteria will not be resistant to that particular antibiotic, right? So that is selectable marker. This is one example. There are more selectable markers. I will tell you that as well. Restriction enzyme, now you know. Probes. What are probes? Probes will help us to identify the particular DNA. Probes are complementary to a particular sequence of DNA. Okay, and they can be radio labeled as well. Ligases. Ligases are the molecular glue. Ligases are molecular glue. That will help us to stick the cut ends of the DNA. So correct option for this question will be C without any exception. Okay. So now let's move on to our next question. Analysis of a chromosomal DNA using the southern hybridization technique does not use. First we will understand what is the southern hybridization technique and then we will come to a conclusion. 4 option you have here: electrophoresis, blotting, autoradiography and PCR. ठीक है, चलो देखते हैं. Understand this. What happens in southern blotting technique? Let's understand one by one. Restriction endonuclease will be used to cut our genomic DNA. So here this is genomic DNA. This genomic DNA is cut with the help of restriction enzymes and we have got fragments of DNA. Then we will perform gel electrophoresis. I told you what is a gel electrophoresis. In gel electrophoresis there will be wells here. There will be wells here, and in this well we will load our sample, right? Since our DNA is negatively charged, it will be attracted towards the positive end of our tray. So this is our positive end. Now our DNA will start moving towards the positive end, which is also known as anode, right? Now DNA fragment that is smaller in size will move further, and the fragment that is heavier will not be able to move far, isn't it? So what will happen ultimately? We will get bands of DNA. Now this band of DNA will now be transferred to a nitrocellulose or nylon filter that is known as blotting, southern blotting. Southern blotting किसका हुआ? DNA का. Okay, and ultimately that those DNA fragments will now be attached to the probes, which is radio labeled, and after dyeing with ethidium bromide, now we can see them in UV light or X-rays, right? So basically southern blotting में क्या involve नहीं है, we have seen the whole mechanism. चार option है आपके पास, electrophoresis that is there, blotting is also there, autoradiography is there, what is not there is PCR. And what is PCR? PCR is polymerase chain reaction. We are not amplifying the sample, it is already amplified. हम मल्टीपल कॉपीज बना ही नहीं रहे हैं हमारे जीनोम के, हमारे डीएनए के, तो पीसीआर हमने किया ही नहीं इन सर्दन ब्लॉटिंग. सो क्या नहीं किया इन दिस क्वेश्चन? करेक्ट ऑप्शन विल बी डी, दैट इज पीसीआर. ओके, अंडरस्टूड? इजी. चलो नेक्स्ट क्वेश्चन पे आते हैं. बहुत ही इंटरेस्टिंग सेशन है ये, मेरे लिए तो बहुत इंटरेस्टिंग है, आपके लिए भी होगा. बहुत सारे क्वेश्चंस पूछे जाते हैं बायोटेक्नोलॉजी से, ध्यान दो सभी बच्चे. विच वेक्टर कैन क्लोन ओनली अ स्मॉल फ्रैगमेंट ऑफ डीएनए? डायरेक्ट क्वेश्चन है. बैक्टीरियल आर्टिफिशियल क्रोमोसोम बीएसी, ईस्ट आर्टिफिशियल क्रोमोसोम वाईएसी, प्लाज्मिड और कॉस्मिड. करेक्ट ऑप्शन इज प्लाज्मिड. छोटे से डीएनए के पीस को कौन एम्पलीफाई कौन क्लोन कर सकता है? द करेक्ट ऑप्शन इज प्लाज्मिड. ओके, commonly used vectors for human genome sequencing are very important question given in your NCERT previous year question. Correct option will be BSc and YSc. Human genome project में कौन से vectors use किए गए थे? BSc and YSc. BSc and YSc stands for bacterial artificial chromosome and East artificial chromosome. Okay, so correct option for this question will be B. You need to remember this because it is repeatedly asked in your exam. Next, the colonies of recombinant bacteria appear white in contrast to the blue colonies of non-recombinant bacteria because of. This also I will explain. This is also a type of selectable marker, very very important. This is also known as selectable marker. And why this is used? Why this is used? When I'm using all these processes of biotechnology, I have introduced the gene of interest, I have exposed my bacteria to the vectors, but now I want to identify that which all bacteria are recombinant and which are not. अब मुझे पता करना है कि मेरा जीन ऑफ इंटरेस्ट कौन से बैक्टीरिया में है और किस में नहीं है, कौन से बैक्टीरिया रिकॉम्बिनेंट है और कौन से नहीं है. देन आई विल यूज द सिलेक्टेबल मार्कर्स. ओके, एंड one example is that I will get different colored colonies of bacteria to distinguish between these bacteria. कैसे क्या सब मैं बताऊंगी आपको, but let's see all these options. Insertional inactivation of alpha galactosidase in recombinant bacteria. Inactivation of glycosidase enzyme in recombinant bacteria. Non-recombinant bacteria containing beta galactosidase enzyme. Insertional inactivation of the alpha galactosidase in non-recombinant bacteria. Correct option is C for sure. Correct option is C. Okay, कि non-recombinant bacteria will have this enzyme beta galactosidase because the gene that is producing this beta galactosidase is not disturbed at all. वो जीन जो बीटा गैलेक्टोसाइडेस एंजाइम बनाएगा वो बिल्कुल भी डिस्टर्ब नहीं हुआ, उसमें कुछ भी हमने जीन ऑफ इंटरेस्ट जो है इंट्रोड्यूस नहीं कर पाए. नाउ दैट्स व्हाई वी विल गेट these type of colonies. Let's understand how. Okay, now this is my plasmid vector. Now this plasmid vector will be inside a bacteria, isn't it? Plasmids are inside a bacteria, so this plasmid vector will be inside a bacteria. Now what I want is, I want to introduce my foreign DNA, which is known as gene of interest. Okay, I want to introduce my gene of interest in this plasmid vector that is inside a bacteria. Fine. Now, I have exposed my competent E. coli bacteria to this plasmid in the medium. In my medium, I have this plasmid and I want my bacteria to take up this plasmid. Okay, and here you can see my competent E. coli is having LAC-Z gene. Okay, now this LAC-Z or LAC-Z gene is responsible for formation of an enzyme which is known as beta galactosidase. So I will write here my LAC-Z or Z gene will form beta galactosidase enzyme. What is the function of this enzyme? That I will tell you. For now इतना याद रखो कि मेरा ये जो LAC-Z जीन है, दिख नहीं रहा होगा, ओके, मेरा जो LAC-Z जीन है वो बनाता है बीटा गैलेक्टोसाइडेस एंजाइम जो एक पर्टिकुलर कंपोनेंट ऑफ मीडियम को ब्रेक करता है. दैट वी विल सी. नाउ अगर if by chance my foreign DNA is introduced inside this gene, okay, so there are three options. In first option, my LAC-Z gene is disturbed because of insertion of this foreign DNA. LAC-Z जीन था, उसके बीच में ही हमारा foreign DNA चला गया है, तो अब ये LAC-Z जीन क्या बीटा गैलेक्टोसाइडेस एंजाइम प्रोड्यूस कर पाएगा? नहीं कर पाएगा. It is disturbed. Insert outside the LAC-Z gene. LAC-Z जीन के बगल में जो है हमारा foreign DNA insert हो गया है. So there is no problem in the LAC-Z gene. Now it will be able to form this beta galactosidase enzyme. Third option is insert ही नहीं हुआ है, anywhere in the plasmid, हुआ ही नहीं है, तो again my LAC-Z gene is intact, वो अपना काम करता रहेगा, अपना enzyme जो है produce करता रहेगा. So there are three options. Okay. Now what will happen when I will grow this E. coli in a culture medium containing X-gal? X-gal is the substance that is broken down by this enzyme beta galactosidase, and because of which I will get blue colonies. So if this enzyme is present, if this enzyme is formed, then only this substance will be broken down and then only I will get blue colonies. अगर वो एंजाइम है ही नहीं, मेरा सब्सटेंस ब्रेक ही नहीं होगा, ब्लू कॉलोनीज ही मुझे नहीं मिलेंगी एंड आई विल गेट वाइट कॉलोनीज. हाय पृथ्वीराज, अंडरस्टूड? बहुत सिंपल. ये जो जीन है दैट इज लेग-Z जीन, जिसके वजह से ये एंजाइम बनता है, बीटा गैलेक्टोसाइडेज एंजाइम। ये एंजाइम ही ब्रेक करता है एक्स गाल को और हमें मिलता है ब्लू कलर कॉलोनीज ऑफ बैक्टीरिया। ओके, और अगर ये एंजाइम जो है, ये एंजाइम अपना बनेगा ही नहीं, क्योंकि हमारे जीन में ही हमने हमारा जीन ऑफ इंटरेस्ट ऐड कर दिया है, तो कॉलोनीज हमें वाइट ही मिलेंगी। So these are the colonies of recombinant bacteria and this is a very good selectable marker. Understood? Okay. Chitin क्या है? Chemically chitin is a polymer and it is a homopolymer of N-acetyl glucosamine. Chitin हमें कहाँ मिलता है? आर्थ्रोपोडा के एक्सटर्नल आर्थ्रोपोडा के एक्सटर्नल स्केलेटन में, राइट? और हमें कहाँ मिलता है काइटिन? फंगाई के सेल वॉल में। काइटिन इज अ होमोपॉलीमर जो कि किससे बना होता है? एन-एसिटाइल ग्लूकोसामीन के कॉम्बिनेशन से। ओके, होप दिस वाज हेल्पफुल। नाउ लेट्स मूव ऑन टू आवर नेक्स्ट क्वेश्चन। आवर नेक्स्ट क्वेश्चन इज डीएनए फ्रेगमेंट्स generated by the restriction endonuclease in a chemical reaction can be separated by, है ना? क्या किया हमने हमारा एक डीएनए फ्रेगमेंट लिया, उस फ्रेगमेंट को जो है हमने कट कर दिया विद द हेल्प ऑफ रेस्ट्रिक्शन एंडोन्यूक्लियस और रेस्ट्रिक्शन एंजाइम, और अब मुझे उसको सेपरेट करना है। सो हाउ विल आई सेपरेट? यस, वेरी गुड शिव। हाउ विल आई सेपरेट? इलेक्ट्रोफोरेसिस से, रेस्ट्रिक्शन मैपिंग से, सेंट्रीफ्यूगेशन या पॉलीमरेज चेन रिएक्शन से। आई विल यूज जेल इलेक्ट्रोफोरेसिस। अभी हमने देखा, आई विल कीप ऑल दीज फ्रेगमेंट्स इन दिस जेल in a well and then it will be attracted towards the anode और वो जो fragment है अपना separate होता जाएगा। Then I will use a staining technique by using ethidium bromide dye। मैं stain करूँगी उस DNA को and then I will be able to view them in UV light। Simple gel electrophoresis। Centrifugation से हमें नहीं मिलेगा। पॉलीमरेज चेन रिएक्शन का मतलब होता है एम्पलीफिकेशन ऑफ डीएनए। अगर मुझे मल्टीपल कॉपीज बनानी है डीएनए की, देन आई विल यूज दिस पॉलीमरेज चेन रिएक्शन। सो सिंपल आंसर ऑफ दिस क्वेश्चन इज जेल इलेक्ट्रोफोरेसिस। ओके, नाउ डियर स्टूडेंट्स, दैट वाज ऑल फॉर टुडे। होप यू ऑल हैव एंजॉयड दिस सेशन। बहुत सारी इन्फॉर्मेशन भी आपने गैदर की होगी, बहुत कुछ आपको समझ में आया होगा, जो भी कंफ्यूजन है आपके क्लियर हुए होंगे। ये सारे प्रीवियस ईयर क्वेश्चंस बहुत इंपॉर्टेंट है। कल भी हम लोग डिस्कस करेंगे प्रीवियस ईयर क्वेश्चंस ऑफ बायोटेक्नोलॉजी एंड इट्स एप्लीकेशन। ओके, तो उसके सारे प्रीवियस ईयर क्वेश्चंस हम कल डिस्कस कर लेंगे, राइट? ऑल्सो एक इंपॉर्टेंट इंफॉर्मेशन देनी थी आप सभी को कि यू ऑल कैन ज्वाइन आवर नीट 22 रैंक अश्योरेंस अल्टीमेट पैकेज। अगर आप नीट 22 के लिए अपेयर कर रहे हो, देन यू कैन टेक दिस पैकेज डियर स्टूडेंट्स। infinitylearn.com/NEET हमारा जो वेबसाइट है, वहाँ पे जाके जो है आप इसको रजिस्टर कर सकते हो। एंड इन दिस यू विल गेट लाइव क्लासेस, यू विल गेट सेल्फ लर्न वीडियोज, सेल्फ लर्न फ्लैश कार्ड्स, अडेप्टिव प्रैक्टिस, you will get daily test and test series and doubt app as well. Okay, what is the price? पृथ्वीराज, प्राइस तो अभी बहुत कम चल रहा है बच्चा। 499 में जो है आपको ये सारे पैकेजेस मिल रहे हैं। कैसे आप उस पैकेज को ले सकते हो वो भी मैं आपको बताऊंगी। एंड ऑल्सो यू कैन ज्वाइन आवर टेलीग्राम चैनल, इन्फिनिटी लर्न का हमारा जो टेलीग्राम चैनल है, दिस ऑल्सो यू कैन ज्वाइन। डोंट फॉरगेट टू लाइक एंड शेयर दिस सेशन एंड सब्सक्राइब करना मत भूलना इन्फिनिटी लर्न नीट चैनल को। ओके, सो ये पैकेज आप कैसे ले सकते हो मैं आपको बताती हूँ। जिस तरह से आप यहाँ मेरा वीडियो देख रहे हो, ओके, आपको जाना है हमारे डिस्क्रिप्शन बॉक्स में, ठीक है? डिस्क्रिप्शन बॉक्स में आपको जाना है, मोर आपको यहाँ पर दिखेगा। शो लेस कर देती हूँ मैं, सो दैट यू विल बी एबल टू सी इट क्लियरली। ओके, लाइक दिस। Okay, like this you will see our description box. Okay, click on show more. Show more पे click करो, you will see all these links. जो ये वाला लिंक है, register here for Infinity Learn free master class, इस लिंक पे आप क्लिक करोगे and you will come to our web page and in our web page you can fill your details here. और इसको आप नीचे स्क्रॉल करोगे तो आपको यहाँ पर जो आप देख रहे हो that 499 में you are getting this package. 70% डिस्काउंट पे सारे सब्जेक्ट्स जो आपके कवर होंगे। यू कैन सिलेक्ट द एग्जाम दैट यू आर प्रिपेयरिंग फॉर, अगर आप नीट के लिए प्रिपेयर कर रहे हो तो आप वहाँ पे क्लिक करोगे, राइट। फिजिक्स, केमिस्ट्री, बायोलॉजी तीनों सब्जेक्ट्स यहाँ पर कवर होंगे। यहाँ पे आपको मिलेगा 3 मॉक टेस्ट, 14 प्रीवियस ईयर क्वेश्चन पेपर, 1000+ सेल्फ लर्न वीडियोस, 1000+ सेल्फ लर्न फ्लैश कार्ड्स, adaptive practice questions and doubt resolution. Okay, and here you can register by submitting your details. So hope dear students this session was helpful. Please don't forget to subscribe Infinity Learn NEET channel, okay, for all the latest updates. Thank you very much. See you in the next class.